All right, student. So let's go back to our continue our lecture. So this is part of our cell physiology. So what we are going to try to talk about is more detail into the protein synthesis. Why is that important? Because as you can see that very beginning of our lecture, we talk about the cell membrane, and the cell membrane has component that is phospholipid, but phospholipid basically block everything. Uh, Nothing can go through unless that's lipid soluble. So you have very limited thing can go through. You will need a protein to do extra work. So protein synthesis is the key of cell function. Without the protein synthesis, you have the membrane, you have the compartment, just like you have the house, but there's no window, there's no door, so nothing can go through, right? So this is not a functional room, right? It's not a functional house. You have to have some window, some door, and those windows and door and a lot of cell function are all produced by providing the protein on the membrane or in the cell. So protein synthesis is the key of cell function. So let's look at what causes the protein synthesis. And the protein synthesis is basically is a way that cell express or present its function. You need to know that every cell in your body come from the same cell. So every cell has the same set of chromosome or DNA. So here is an example of the chromosome in your human cell. So you have 23, 1, 2, 3, 5, 6, 10, 22, 23. 23 pairs of chromosome. Why is a pair? Because you, the origin of you, this individual, come from a cell, and that cell is a combination of one sperm and one egg. Your mother provide the sperm, sorry, your mother provide the egg, your father provide the sperm. Sperm has 23 chromosomes. Egg has 23 chromosomes. So these 23 merge together to form 23 pairs of chromosomes. So that's where you're from. So basically each chromosome, you have one from your mother, one from your dad, one from your mother, one from your dad. They are very they have the same set of coding. There are some variations, but they have these codings are used to serve the protein synthesis. So you can kind of imagine that. For example, what kind of protein do you have in your body? What protein, right? You have a protein in your body, but what are the proteins in your body? A very common protein, later we will talk about, are, for example, digestive enzyme, right? You have the amylase secret from your saliva, which can digest carbohydrates. So that enzyme, that protein, is one of these code. One of these code. It could be in the chromosome 3 or 4 or 11, 17, etc. It could be every protein come from one specific gene, and that gene is located in one of these chromosomes. So you kind of can imagine that you have two genes, and those two genes work. If one gene doesn't work, the other gene hopefully that work. So if your father just happen to don't have the gene to code for amylase, and if your mother does have it, then you do have it. So you still can produce the amylase. Insulin, for example. That's a protein probably everybody knows about. It's an enzyme. It's a hormone to facilitate glucose metabolism in a way, and so insulin comes from one of the chromosome. If the father doesn't have that gene, most people has it, so it's not happening. Oh, there is some situation that people don't have the insulin um, uh, or some other gene, right? So it will be some deficit to produce those proteins. And but if mother does have it, then the person still can produce those proteins. So this is how we produce protein. We basically produce protein from the gene expression. The gene are here. So where are, again, location and function, chromosome, all that's all good. Where are they? They are in the cell. So they are located, where is that? Where is that? Let's see. They are located in, let me see, let me show you. They are located in the nucleus. So in the beginning, you have single zygote. That is the origin of your body development. And then it divides, 1 becomes 2, 2 becomes 4, 4 becomes 16. So it divides. And then once it divides, it's still the same cell, right? Each one will start to differentiate. Some of those become the heart cell, some of those become muscle cells, some of those become the brain cells. So those are but they all come from the same cell. So they have the same set of chromosomes. When these chromosomes, when the cells are going to produce its function, for example, they want to have a protein to anchor on the membrane. That's just one example. They can produce different kind of protein. Once these cells is in a situation that they need to perform its function, then it will need to have protein. So it will start to look into the gene and to activate the gene that they need to produce those proteins. So let's look at how does gene does this protein synthesis. We call this protein synthesis process a gene expression. Gene expression is that there are thousands of different genes, but not every gene will get expressed. For example, red blood cells, the blood cells in your blood, right? Red blood cells, they will not produce keratin. Keratin is a protein we only see those, we commonly see those in your skin. They are from the same cells. They are from zygotes, but they have the same set of chromosomes, but they don't express. Red blood cells don't express keratin. Skin cells don't express the gene to produce hemoglobin, right? So different cells will express different genes. Now, when the cells are in the situation that they need to express to produce protein, they will do gene expression. So let's say that this set of gene will get expressed. So the gene expression process are the process to produce protein. So what are the process? They basically go through two processes. One is called transcription, and then it's called translation. Location and function, transcription occur in the nucleus. So this is DNA. This is the RNA polymerase. This is enzyme. When the gene is going to get expressed, RNA polymerase attach on that gene, and they will read the DNA code and they use it to transcribe the RNA. It will be pre-mRNA. This M refers to messenger RNA. You'll first produce pre-mRNA, produce the mRNA, then these mRNA will travel from the nucleus into cytosol or cytoplasm, and into the cytosol, you will see ribosome. Where do you have the ribosome? We have two types of ribosome. Some of those are attached to endoplasmic reticulum. Some of those are free. So they will reach the ribosome, and then ribosome will do translation. They will use this mRNA code to produce the amino acid polymer. So this one, each of these dots is an amino acid. It will combine together to form a polymer, and this string will be the first structure, the first product. It's not mature yet. This is not a complete product yet. This is a polymer of amino acid. We call it a polypeptide. This polypeptide can be long, can be short, but this is just a string, right? Just a string right here. So this is the basic two-step of gene expression to produce the very first immature products of the protein. So what do you need to know? You need to know these two steps, transcription and translation. And again, location and function, what does it really mean? Transcription is to use the DNA to produce RNA. And where is it? It's in the nucleus. And then translation, where is translation? Translation occurs in the ribosome. Ribosome basically is in the cytosol or cytoplasm. Ribosome can be attached or free. And then what does it do? It will use the RNA code to produce amino acid polymer. So that's those process. So transcription, we talk about that. Basically, those are the very first step of the gene expression. And every cell has the potential to express every protein that you need in the body. So every cell has that potential, but not every gene will get expressed. It really depending on the function of that cell. So on the other hand, to see it is that, another way to see it is that, how do you know the function of the cell? Look at the protein they produce. So if they produce a certain type of protein, you know that's the function of those cells. So that set, we call it differentiation process. This cell will gradually become a particular functional cell, differentiate, separate it from brain, it's different from the heart, different from the blood, different from the skin. Why is that? Because they are differentiated. They produce different set of protein. So the very first step is gene expression. Sorry, the very first step of that protein synthesis is transcription. The very first step of gene expression, transcription. What does it do? Convert DNA into RNA. These, because this is very unique to that cell. These determine the function of these cells. So this is very highly regulated. A cell will do that gene expression only when these cells are located in particular location, anatomical location, and also at the right time, right? So for example, only the cell become located in the skin, they will start to produce keratin. And some of those will become, the keratin will be either from the skin or from the nail or from the hair. So it's different expression will occur by different environmental stimulation, environmental condition. And so this environmental condition can be basically summarized by these protein called transcription factor. This one regulates the gene expression. And then you will also need another enzyme, which is called RNA polymerase. So whenever you see something like ACE, you know it's an enzyme. It facilitates a chemical process. So with these two, they form a complex, and they will bind on a gene location is called promoter, and from those region, these RNA polymers will move along the DNA and to synthesize the RNA. So this is just an example that DNA is a double strand DNA. You have a one strand here, the other strand here. RNA polymers attached on it. They will first attach on the promoter region, and the gene transcription factor recognize those promoter region, call on the RNA polymers. These two come together and go along this DNA, and then it will produce the RNA. So this is how we produce the RNA. Where is it? It's the nucleus. What are the players to work together to form it? You got to have DNA, right? You got to have those genes. You got to have RNA polymers. You also need transcription factor that represent the environmental stimulus, environmental condition. So different environment will provide different transcription factor. If the transcription factor is messed up, you will have very weird development, right? You got to have hair only in the skin, but not in the heart, right? So you cannot have the muscle cells to produce keratin. That will be very strange. So you got to have very good, you got to have a correct transcription factor in that environment in order to push the cells to express those proteins. So this is example that here is a nucleus. This is nucleus, and then you can see that this is DNA transcription to form the RNA. RNA will do some process. This is called alternative splicing. It has the exon, intron, remove the intron, combine with the exon, intron removal, do some processing, and these will form the messenger RNA. Then these RNA will then move out. You can see that these one messenger RNA move out, leave the nucleus. Where do they go? They go to ribosome. So next stop is the ribosome. And then ribosome so here is the RNA. So this is the RNA code. I can show you the ribosome. Ribosome is this is an example of the ribosome. Here is the ribosome. Where is the ribosome? Let me show you here. Let me see that. So ribosome is here. Go back to the nucleus. You can see that these dots, these here, you can see that ribosome right here, ribosome. Yeah, these are the ribosome. You have some fixed one in the rough membrane. You also have some free one, just floating around. So you have this ribosome. So after transcription, RNA flow out of the nucleus, the next step is to go to the ribosome. And from the ribosome, here is the RNA. Ribosome will use this code. Ribosome will read this RNA code. It will have a GCAU, etc., right? And the ribosome will read it 3 code as a unit. We call this a codon. A quick question can be that how many RNA nucleotides are used to form a unit for the amino acid coding. That would be 3, right? Three of them bind together as a codon. Then each one, so this is a code, and then RNA will go through this table. For example, GCU, it will find GCU, it will form the alanine, and so the amino acid will form here. Another amino acid will form here, from here. From here, you can translate. What is a translate? Translate is for example, you're gonna translate English to Japanese, for example, is different text, right? Different words. You translate from the nucleotide into amino acid, from this code into amino acid. And so you will translate these RNA code into another sequence, and that sequence is formed by the amino acid polymer. And so that's how we do it. That's how ribosome does it. So here is showing you that ribosome will go along with the messenger RNA, and it will read the code. 3 code as a codon. It will form these. These are the amino acid. And because while it read it, it will generate, it will pop out an amino acid, pop out an amino acid. And so this is the amino acid. There are some chaperone protein, just keep them, in together. And then in the end, really finish it, you have a sequence of the amino acid. This is just the very beginning of the protein synthesis. But now at least you have the material, you have the protein. Where is it? Is in the formed by the ribosome. This protein has 2 directions. It can either packaged in the endoplasmic reticulum, or it can freely diffuse into the cytosol. So they have depending on the destiny of this protein, they will either intracellularly or they will packed in the vesicles. So that's that. So here is the sequence, the very first sequence of the amino acid polymers, and we call it peptide or polypeptide. So this amino acid form a peptide or polypeptide, or if it's pretty long, it's protein. But usually we don't call this protein yet, because this is just a string. This string doesn't have a function. Okay, this string just provide you the material, but this is good material. So what we are going to do, what cells are going to do, is to do folding. So very much like you have piece of paper, you start to fold it. You can fold it into the airplane. You can fold it into an animal. You can fold it into a different like a toy, right? So you can fold it. And we will do the same thing. We will start to use this to fold it. Usually we fold this thing in the Golgi apparatus, but basically this is the first material. So we call this a linear structure or primary structure of the protein. Then this is not functional, we will fold it. We will fold it into 2-dimensional secondary structure. 3-dimensional, you can fold it, and it can be like a linear structure, and then fold it into the 2-dimensional secondary structure, or 3-dimensional to be very like a 3-dimensional, right? And then you can have a 2 or 3 peptide put together as a quaternary structure. So that is how we do protein folding. So here goes some detail here. Primary structure is just a linear structure, so it's not functional. We can have a secondary structure, so it can be alpha helix. Let me see if I have a structure here. For example, these are alpha helix. So this is secondary structure. Yeah, secondary structure. You can also have a beta pleated sheet. So these string can go and then form like a piece of paper, right? A sheet. And also you can also use this piece of paper, then further fold it to form a tunnel. For example, we have this channel, right? For example, this channel. This channel is very much like a straw, right? A straw right there. How do you form straw? If you cut the straw open, it's open up like a sheet, right? So it's like a paper, but you can fold it to form a straw, and you basically just stuck that straw here, and it become a channel. So this is 3-dimensional. So you can fold it as a 3-dimensional. Or you can have 2 or 3 peptide bind together to form a quaternary structure. It's very easy to understand. For example, you have what can I think about? So for example, you have a pencil, a pen. How about that? You have a cap and you have pen, right? You put together, or you have a water bottle, right? Your water bottle, you have a lid and a bottle, and those two are separated. They are from different like gene expression, different protein, but you put them together as a finalized product. Without the cap, water bottle will be liquid, right? You got to have a cap to close it. So that will be the quaternary structure. So that's how we form the protein. Now you have a functional protein. So this is an example of our quaternary structure, the final product of a protein. This protein is in this case, this protein is a hemoglobin. So what do we have here is that you can see that this one heme here, don't worry about the heme yet, but you can see that these are protein. These are the sequence. If you just untangle it, you see it's a linear structure. But that linear structure is not going to do any work. You got to fold it. So first is that you have the secondary structure. This is the alpha helix, right? That's one of the secondary structure right here. Right here, alpha helix. So you have the alpha helix. So make it like a little bit thicker, but still like a string structure. But with this alpha helix, it's very much like yeah. So and then it will fold together. With that, you form a pocket. It's very much like a knitting. Do you guys do knitting? Knit together, and then you can knit a glove. You can knit a beanie. You can knit a vest, right? Then you knit a pocket. This pocket is important to host a unit called heme unit. This is important for our oxygen carriage, so it can carry an oxygen here. And you have this one here. That's good enough, right? That's pretty good. You have a pocket. I will say that is functional. You can have you can host a heme here. You can carry oxygen. I would be impressed if anyone can do that with their knitting. Can you do it? I cannot. My girls, my daughter can do it, but it's very impressive. So you can see that they knit together and form a little pocket, right? And so that's all good. But our body will do even more. You can understand that our body is human body is very complicated. It's very sophisticated. So our red blood cell has this one, but they are not satisfied. They want to bind 4 together. So now this protein, this final protein has 4 of these. 1, 2, 3, 4. 4 of these. Now each protein with this one is even more powerful. Each one can carry 4 oxygen because each one can carry 1 oxygen. Now you have 4. You can carry 4 oxygen. So you have more flexibility. This is like very basic bicycle, right? You can only carry 1 customer. You ride your bicycle, you can only carry 1. But now you got upgrade to an Uber, right? Each taxi or each Uber, you can carry 4 oxygen. So you get upgraded. This is what we have in our red blood cells. Each red blood cell has a lot of this hemoglobin. I got it, hemoglobin. So this term. It's a globular protein. It's a round globular. It has 4 chain. Does it have a chain here? So it's called chain. Let me write it. Sorry, 4 chain. So it has 1, 2, 3, 4, 4 chain, and 4 chains, right? So each chain is basically a separated protein. So you can kind of imagine that one hemoglobin is a quaternary structure. So by knowing it, you know that it doesn't come from one protein, one peptide sequence. It comes from four or multiple, right? In this case, it's four. And so you can imagine that each one, if you go back, each one of these chain is a folded product from one gene expression. And because it has four chains, you can imagine that it comes from four gene expression. It comes from four different genes. So if you go back to the DNA, you can imagine that one hemoglobin has to come from four genes located in this chromosome. There are diseases that for some people, one of that gene is defect. And so the end product, you will have three of them probably okay, right? You have three of them probably okay, but one of that come from a defect gene. So the chain will not function perfectly, and so it will have some oxygen carrying deficit. The person will have less oxygen capacity, and so that will be anemia, right? So that will be the disease that the blood cannot carry enough oxygen. That's called anemia, like lack of the oxygen supply into the cell. Why do you need oxygen? Because your mitochondria, right here, your mitochondria right here, mitochondria need oxygen to do aerobic respiration, to use that oxygen to do oxidative phosphorylation to produce ATP. So you got to have oxygen. You got to breathe in oxygen. You got to use blood to carry that oxygen to the mitochondria, and so they can produce the ATP. So that's protein synthesis from the very beginning to the end. Let's see if I have a picture. This is the final product, the quaternary structure, and so this is ribosome. This is ribosome. So ribosomes, you can have a free ribosome that is in the cytosol, and also membrane-bound ribosome. It attached to the endoplasmic reticulum, and then ribosome will, what does it do? It will convert. It will do the translation. It will convert the mRNA into protein, right? Or amino acid polypeptide. And then this, right here. So if it's free ribosome, it will just release this one into cytosol. But if it's fixed ribosome, if it's membrane-bound ribosome, right here, ribosome attached to the rough ER, it produces the peptide. This peptide is not going to freely diffuse around. It will get packed into the endoplasmic reticulum, ER, and then it will get packed here, folded, packaged into the vesicle. So this vesicle was a protein, was a material in this membrane, is a lipid. So it will close it, transport it. It will transport it to the next step. That is Golgi apparatus, right here. So here is the map, right? So this is the nucleus. Here you have the chromosome, and then attached to next to it is endoplasmic reticulum. Here is the ribosome. Ribosome will then convert the RNA to protein. When it produces protein, it will pack it into the vesicle, then it will transport these into Golgi apparatus, what does Golgi apparatus do? Golgi apparatus will fold these protein. You have linear structure, you gradually fold it into secondary structure to tertiary structure to quaternary structure. Then you form the final product. Golgi apparatus, in this case, it will show, this one show you that we can transport, we can release this protein into outside of the cell. This is important for cells function. For example, some cells will release their protein. For example, insulin, they will produce the insulin. Insulin will floating around. It will help you to conduct this glucose metabolism. Yeah, and that's what we talk about, right? We talk about the beta cells and insulin when we talk about the glucose homeostasis, right? So this is protein. This is protein synthesis. This is how we travel. So one thing you need to know is that every cell has a potential to produce different kind of protein, but they don't do that. We don't have a red blood cell to produce keratin. We don't have the eye cells, right, to produce hemoglobin, right? So different cells produce different protein. However, every cell has the same set of chromosomes. They have potential. They just don't differentiate to, they all differentiate into different cells. And so how do you know the function of cell? Look at their protein. See what proteins they produce. And that determine their function. One of the protein, sorry, here just show you. Some of the protein are released. Some of the protein are attached to a membrane. So this is a membrane protein. We talk about the ion channel. We talk about the pump. Those are membrane protein. You come from the DNA, RNA, translation, folded, package here, and then either attached to a membrane, move it here, attach to the membrane, or release that, right? So you can have a membrane protein or secretory protein. You can also have some proteins stay in the cell. Hemoglobin is the example. They stay in the cell. They stay in the red blood cells. So that is proteins. They are the media. They are how cells produce their function. You can have a signal. This will be signal, insulin, for example, something like that. It will act on it to trigger the cell to do different work. So this one give you the idea that even though every cell comes from the same origin, this is the very beginning of your life. The oocyte, the egg from your mother, the sperm from your father, merge together. These form the very first cell, zygote. It has 23 sets of the chromosome. Then it will divide. But you will grow into a different organ system. Different organ system gonna develop into your body. Then the cells in those environment will get different environment stimulus, different transcription factor. Then they will start to express different gene. When they express different gene, they will have different kind of cytoskeleton. Their structure will be changed. They will produce different protein. They will produce different function. So you can have a nerve cell. You can have a skin cell. You can have a muscle cell, blood cell. You can have a different organ cell, digestive system, liver cell, pancreas cells, right? Even though they all function different, they all produce from different protein, produce different protein, they have the same set of the chromosome. And so a very important concept to know is that the protein production determine their function. Cells produce different protein, and that determine their function. Every cell in the beginning, they are just like baby. Every one of you has the different potential. And how do you know that you do your job? How do you know that you are mature to perform your calling or your profession? Look at what you produce, right? Look at what cell produce. If the cell produce a particular protein, we know that is a functional, differentiated, matured cell. Otherwise, they will be stem cell. They are original cell. They don't have particular function. Their only function is just get divided, right? Multiplied. So that's that. We call this cells function, cells phenotype. Their function get observed, right? This is a process called cell differentiation, and just like every one of you, just like every kids, right? Every kid has a potential. But how do we know that they perform their function? See what they do. If you are a doctor, right? You have to produce that function to take care of your patient. If you are a mother, you produce that function, you produce that fruit, you produce that outcome to take care of the baby, something like that, right? So yeah, I'm a teacher, so what I'm doing is to help my students. How do you know I'm a teacher? To see what I do, to see what I produce, basically. Yeah. So connect this one with the membrane. So this is the membrane protein. Let's talk about the membrane protein later, but keep in mind that all these membrane proteins are protein products from what? From gene expression. Okay. All right. So let's take a break, and we will continue with the membrane protein, because from here I want to build the foundation for electrophysiology. It's very easy, very straightforward, but let's take a break and we come back.